Niacin's protective role in atherosclerosis extends well beyond its impact on lipid metabolism. It also exerts a direct anti-apoptotic effect in immune cells, particularly macrophages exposed to oxidized low-density lipoprotein, OXLDL, a key driver of cellular stress and plaque progression. In the hostile microenvironment of the atherosclerotic lesion, where oxidative stress, inflammation, and lipid overload converge, macrophage viability becomes critical. These cells not only help clear lipids but also regulate immune responses, tissue remodeling, and resolution of inflammation. Niacin, through a multifaceted mechanism, preserves macrophage integrity and function, reducing the formation of necrotic cores and improving plaque stability. When macrophages ingest OXLDL, free cholesterol accumulates within lysosomes. This overload disrupts lysosomal membrane integrity, triggering the release of cathepsins and activating inflammatory signaling pathways, including the NLRP3 inflammasome. Simultaneously, cholesterol crystals may form, further damaging organelles and leading to mitochondrial dysfunction. These events culminate in caspase activation and apoptosis, an outcome that not only depletes the macrophage population but also contributes to necrotic core development and increased plaque vulnerability. Niacin interrupts this cascade at several points. First, it enhances the expression of CD38, which catalyzes the production of nicotinic acid adenine dinucleotide phosphate, NAP. NAC mobilizes calcium from lysosomes via TRPML1 channels, a signal that activates the transcription factor LXRA. This, in turn, upregulates critical transporters such as Niemann-Pick C1, NPC1, ABCA1, and ABCG1. These proteins work together to shuttle free cholesterol out of lysosomes and ultimately out of the cell, preventing the toxic buildup that leads to cell death. By restoring lysosomal cholesterol homeostasis, niacin preserves the integrity of this key organelle. With reduced lysosomal stress, there is less inflammasome activation, less mitochondrial damage, and less triggering of the intrinsic apoptosis pathway. Experimental data confirm this effect. Macrophages treated with niacin in the presence of OXLDL exhibit significantly lower rates of cell death, as assessed by flow cytometry and viability assays. This rescue effect is lost when CD38, LXRA, or NPC1 are silenced, underscoring the importance of this calcium and cholesterol efflux pathway. Importantly, this anti-apoptotic effect has broader implications for immune cell function. Macrophages that survive in an OXLDL-rich environment can continue to participate in lipid clearance and resolution of inflammation. Their presence supports a more regulated, less inflammatory plaque phenotype. Conversely, when macrophages undergo apoptosis and are not efficiently cleared, they release cellular debris and pro-inflammatory mediators, exacerbating lesion progression and contributing to the destabilization of the fibrous cap. Furthermore, niacin's influence on mitochondrial function, likely linked to its support of NAD, metabolism, and sirtuin activation, may reinforce cellular survival mechanisms. NAD is essential for DNA repair, oxidative stress management, and mitochondrial homeostasis, all critical processes for macrophages under atherogenic stress. By enhancing NAD availability, niacin may help these cells adapt more effectively to the metabolic and oxidative challenges posed by OXLDL. In essence, niacin preserves macrophage survival by targeting the root of the stress, lysosomal cholesterol overload. Rather than merely suppressing downstream effects like inflammation or apoptosis, it addresses the initial imbalance, allowing macrophages to maintain function and viability within the atherosclerotic plaque. This contributes not only to a lower necrotic burden but also to a more stable, less rupture-prone lesion. Taken together, these findings position niacin as a powerful modulator of immune cell fate in atherosclerosis. Its anti-apoptotic actions are not incidental but central to its ability to reshape the cellular landscape of plaques. By rescuing macrophages from OXLDL-induced death, niacin supports a healthier, more resilient immune environment, one better equipped to contain lipid accumulation, limit inflammation, and promote long-term vascular stability.